Topic: Genetic mutations. Number of us 1. Key issues. Topic plan. First, genetic mutations. Second, mutations more variation. Third, chromosome mutations and gene mutations. The purpose of the lecture: study the genetic mutations.
In the past two lectures, we discussed the concept of a gene mutation, and we examined the different types of gene mutations that exist in nature. Now, let's actually summarize our results, and let's begin by looking at the following flowchart. So, this flowchart basically describes the many different types of mutations that can arise in nature, and it also tells us the reasons for our gene mutation. Now, a gene mutation is basically a change in the nucleotide sequence on the DNA that is not a result of genetic recombination. Now, basically, there are two reasons for a gene mutation in the first place. So one of these reasons is a result of an error that might take place in one of the many processes that take place in the body. For example, one process in which an error can basically lead to a gene mutation is DNA replication. So during DNA replication, our DNA polymerase can basically make a mistake and incorrectly base pair a certain nucleotide pair, and that will create our gene mutation. So these types of natural gene mutations that take place as a result of one of the many natural processes that exist in the cell is known as a spontaneous mutation. Now, on the other hand, a gene mutation can also take place as a result of one or more physical or chemical agents known as mutagens. So these are basically outside physical or chemical forces that can basically induce our gene mutation. And one example is UV radiation. So UV radiation is an outside physical agent that can basically cause harm and create a gene mutation in DNA. So we have induced mutations or spontaneous mutations. So we have two reasons for our mutations to arise. Now, once we form the mutation, there are two categories of mutations. We have insertion and deletions, as well as point mutations, which are also known as base pair mutations or base pair substitutions. And let's begin by discussing insertion deletions. So to insert something or to delete something means we're basically either placing nucleotides into the DNA or we're removing nucleotides from that DNA sequence. Now, an insertion or deletion of a number of nucleotides that is not a multiple of 3, that is not a multiple of the codons that exist in the genetic code, will cause the reading frame to shift, which will change the amino acid sequence of the polypeptide chain, and this is known as a frame shift mutation. So insertion deletions can form frame shift mutations in which the entire reading frame, the entire mRNA sequence is shifted, and that means that completely new codons will be read by our ribosomes and a completely new sequence of amino acids will be produced. And this usually leads to non-functional proteins. Now, let's take a look at the other type of insertion deletion known as a non-frame shift mutations. So on the other hand, an insertion or deletion of nucleotides that is a multiple of 3 will simply insert or remove a number of amino acids equaling to the number of new codons that are added or removed. And this is known as a non-frame shift mutation because the reading frame is not actually shifted and because the majority of the amino acid in the sequence is actually unchanged. Now, let's move on to the second category known as point mutation. So a point mutation, also known as a base pair mutation or a base pair substitution, is basically a change of a single nucleotide on that DNA template. So if the point mutation does not actually change the amino acid that is produced, and in this case it is known as a silent mutation. Now, a silent mutation can either arise on the non-coding region of the DNA or on the coding region of the DNA. And the reason a silent mutation can arise on the coding region of the DNA is because our genetic code is degenerate, and that means more than one codon can basically code for the same exact amino acid. So if we have a point mutation that causes that forms a new codon, and that new codon still codes for the same exact amino acid, then we know that this is a point mutation because the same exact amino acid sequence will form. On the other hand, a point mutation that changes the codon and the amino acid that is produced is known as a missense mutation. So we have two types of point mutations. They can either be silent or they can be missense. Now actually, all point mutations are also non-frame shift mutations. So let's put that in parentheses. So non-frame shift. So all point mutations are non-frame shift mutations, while insertion and deletions can either be frame shift or non-frame shift. Now, the final type of mutation that I want to briefly discuss is a nonsense mutation. So a nonsense mutation can arise as a result of insertion deletions or point mutations. So both of these categories of mutations contain nonsense mutations. So a nonsense mutation is a mutation when we basically change a codon that codes for an amino acid into a codon that is a stop codon that terminates our polypeptide chain, and this basically terminates the polypeptide chain prematurely, and this causes or creates a non-functional protein. So basically this concludes our discussion on genetic mutations. We have two different types of categories. We have insertion deletions and point mutations. Now, insertion deletions can cause frame shift or non-frame shift mutations, but point mutations themselves are always non-frame shift mutations, and there are two types. We have silent mutations as well as missense mutations, and both of these categories can contain nonsense mutations.
Answer the test questions in the online class. Watch the video on the YouTube and send task to the university system. Thank you for your attention.